Okay, let's get going. I don't want to fall behind schedule. So we have our final presentations. I hope that doesn't mess up the machine. Our final presentations today. We have 1, 2, 3, 4, 5, 6, 7 presentations. So we have just enough. If we have 10 minutes, if we can keep on a 10-minute schedule. If we can keep on a 10-minute schedule, then we'll be in good shape. No. Hey. Hi. Oh, it's still on mute. Okay, got it. Thanks. All right. So let's get going. So Arthur, come on down. So can I put this on you?
Sure. Keep this in your pocket, or
yeah, that's good.
Is there like a pointer or anything I can use, or usually just use hands?
I use my hands, yeah.
Okay, that's fine.
Go ahead.
All right, so hi everyone. My name is Arthur. I'm in the neuroengineering and systems biology track, so I'm interested in genomics. I'm going to be talking today about genomics and signal processing. So just to introduce what genomics is, I'd say it's loosely using computational tools to analyze DNA sequences. And it's been recently enabled by major advances in sequencing technology. So it wasn't really possible to sequence DNA up until about 30 years ago. And we only got the full human genome sequence about 10 years ago. So this is a pretty new field, but it's a source of major investment and wide applications in medicine and biotechnology. Some of the problems in genomics, the one I'm going to be talking about today, is discriminating coding from non-coding regions in DNA. So if you're not familiar with DNA, it's composed of introns and exons. Exons actually encode proteins. So proteins are the things in your cells which do work, and they're encoded in your DNA. The tricky thing is that exons aren't continuous on DNA. So you get exons that are split apart by introns, and they're not really split apart in any kind of predictable way. So it's difficult to figure out which parts of DNA are exons and which parts are introns. And so I'm going to be using signal processing techniques to try and accomplish this task. Within exons, there are these things called codons. These are really just groups of 3 nucleotides. So DNA is made up of nucleotides A, T, C, and G. And so each combination of 3 nucleotides encodes 1 amino acid, and proteins are made up of those amino acids. So I know that's like a really quick crash course in molecular biology, but hopefully that will suffice. So what I'm going to do now is try and apply some signal processing techniques to try and figure out which regions are exons, which regions are introns. So the first thing we need to do is digitize a signal. So you can download DNA sequences from NIH. It's just a long string of A's, T's, C's, and G's. I'm going to digitize in probably the simplest way you can, just defining 4 different unit sequences. Each sequence, mu T, mu A, mu C, mu G, is 1 wherever the sample is T, and 0 everywhere else. So for this example sequence here, A, T, C, G, G, T, A, G, G, A. We have mu T as 1 wherever there's a T and 0 everywhere else. Same thing with A. It's pretty straightforward. Once we have those sequences, we can start calculating the DFTs and start looking at them in the frequency domain, which is sort of counterintuitive because there's no time aspect to this. But it still gives you a sense of the periodicities within the strand, which is useful, as we'll see later on. So I'll define the DFT of mu A to be UA, and the same thing for UT, UC, UG. And within this field, people tend to represent the total spectral content of the sequence as SK, and it's just the square of the sum of the magnitudes of the DFTs. So we can calculate DFT of some length. I use 351 and look at a spectrogram of the sequence. So on the y-axis here, This is the frequency k again. Sort of, you have to think about it just as and each frequency k corresponds to a period of n over k within the sequence. And we're looking at the power as color, and this x-axis here is the sequence location. So the top one is a real sequence. So this is a sequence that actually contains exons. The bottom one is a random sequence. So I just generated them randomly, generated the nucleotides randomly. There shouldn't be any protein coding regions. The top and bottom, there's sort of a lot of mishmash of signal. It doesn't really contain any positional information about where the exons and introns are. What I'd like you to focus on is this band here. So you can see these three red dots here. Kind of hard to see, but if we zoom in, I think they're pretty clear. These actually represent the exons. So in this sequence, there are actually exons at, around these three regions here, and at this fourth region, which is hard to see. And there's actually a fifth one, but the spectrogram didn't pick it up, so it's not perfect. In the random sequence, you can't see any clear distinctions. So what's important is that these bands are located at the frequency of around 117, and I calculated this DFT with 351 points, and so importantly, this is at the n over 3 frequency. So that means that it's periodicity 3. So this is a really important frequency. I guess since the 80s, people have noticed that exon regions tend to have peaks at n over 3, at the frequency n over 3. The intuition behind this, I'm not sure if they figured this out before or after they saw the peak, but codons are used with unequal frequencies in exons. And a given base tends to be located at identical codon positions. So that means that the A, if it's in codon 1, will tend to be at codon 2, at the first region of codon 2 and at codon 3. The same thing with the other nucleotides. So it gives some periodicity to the coding regions. So now I'm going to try and simplify this a little bit and only look at this n over 3 frequency. So it's that where we had k at 117. I'm going to define this this parameter w. You can think of it as it's sort of similar to sk, except we've gotten these optimized parameters, little a, little t, little c, and little g, which they're sort of using a training set to optimally discriminate coding from non-coding regions, but I don't want to dwell on them too much. So now we're only looking at one frequency here. So we're only looking at that n over 3 frequency, looking at the power on the y-axis now, and the region location is still on the x-axis. So that's still, the sequence location is still the same. The x-axis is still the same for all these figures. Again, on the top bottom, we have a real sequence with actual exons and a random sequence. And this is, I think, much clearer than the spectrogram. You can see that we have these 5 nice peaks corresponding to the coding regions of the DNA. And this was actually, these figures were reproduced from a paper, which was only looking at this sequence. And so the paper shows these exons really nicely. What I was curious about is how well this parameter W would be in actually predicting where other exons are. So I wanted to try and use this method on some new sequences. And so what I did is we have the old sequence here. So that's about from here to here. And then I took the sequence and pushed it out much further. So I looked much further in the genome. So before we were only looking at about 8,000 bases. Now we have 40,000 bases. And these pink bars here, these are where the actual exons are. So they were experimentally determined. And so the blue signal, what you see, is the same thing. It's a power, except for a longer sequence. These dots up here on the top, these are where I actually predict, based on this based on the power here, or the value, or the magnitude of w squared, where the exons are. So where you have these strings of blue dots, that's where I predict where the exons are, based on a very simple algorithm. I'm just taking a threshold and then saying, well, an exon has to be a minimum length. I'm saying it has to be 50 nucleotides long, which is biologically pretty accurate. So the tricky thing is, where do you set this threshold? That's a pretty typical problem with these kind of things. We can see that for these five exons that we showed in the graph previously, this algorithm works pretty beautifully. It finds the exons really well. Finds some other ones pretty decently too. So like here. So where these blue lines are overlapping with the salmon colored bars, that's good. Problem is we're missing a lot too. So it's not perfect. But it does give you, and also the edges aren't clear. So we're not only finding, we're not finding the edges of the exons perfectly, but they're pretty close for when it works. If we want to try and find more of the pink bars, we can reduce the threshold at which I define, okay, this is an exon. Trouble is that the further I reduce the threshold, the more junk I'm going to be picking up. It's not actually exons, but I will also be picking up a higher fraction of the exons. So it's a trade-off. I can also change the STFT window. So I used 351 before. You could try making the window bigger. You might get higher powers, but you will also start to find more junk, I think. And you run into the same problem, into the opposite problem if you reduce the window size. So if you would go with a smaller window, you'd be able to find smaller exons, but you'd also have lower SNR. So you have to use a lot of trade-offs. But ultimately, I think this technique is pretty useful in finding at least the general area of the exons, even though it's not perfect for finding the exact locations. So yeah, I think it's a useful tool in genomics, and you can use further downstream processing using more conventional biological methods to more precisely find the exon. So thanks.
All right, thanks. Okay, we need to move on because of the timing, but that was great. Thanks a lot. So, yeah. Yeah, sure, if you want to. So, your slides on here?
Yep.
All right. So, Dongshui, go ahead.
Yeah. Today we're going to talk about blind signal separation. My name is Yue Hou, and this is Dongshui. And our team name is No. These are what we're going to talk about today. Include a brief introduction of blind signal separation and a problem specification of our project and result analysis. Dongshui will talk about result analysis and the future improvements and conclusion. As an introduction, imagine you were in a party and you were talking to someone else, and how would you recognize another person talk to you? That's what blind signal separation do. Basically, it will separate a mixed signal with several individual signals mixed together, and it helps people recognize later in maybe a recorder. It will separate the individual signals. And there are several applications for the blind signal separation, such as acoustics and conference SSI. When you record a conference, there are a lot of people speaking together, and after that, you may want to only focus on one person's idea, and you may probably need it. This is the famous cocktail party problem. It's the basic of the blind signal separation. It's just, as you can see from the left, there are several individual sources, and in the middle of it, you can just regard it as recorders. And each of the recorder has all the signals together. And after that, this part is the blind signal separation process. middle right, those are the outputs of the blind signal separation, which is just as the previous original signals. And in our project, we are trying to separate from 3 original signals we find online, and use a random matrix in the MATLAB, generate in the MATLAB to mix the 3 signals together, and use our method to blind signal separation method to separate the mixed signal into 3 outputs, which will be very similar to the original signals. And next, I will pass to Dongxue to talk about the algorithm.
Okay. The algorithm we adopt is ICA algorithm. It is actually a statistical algorithm. And the goal of this ICA is to get a linear transformation, which is the transpose of W, that can maximum the statistical independence of the component of the observed signals, which is X, and X is a mixed signal. The process of this algorithm is here. Firstly, we should make the mean value of the source signal to 0, and this won't change the statistical features of this matrix. And next, we should do the data whitening, which would improve the possibility of the convergence of this algorithm. And the next step to select a random orthogonal matrix W0 as an initial matrix before the iteration. And then we start the iteration and find the W that can get the maximum value of the matching using this formula as follows. And since we have adopted this algorithm from statistics, we should also try to process the signal using DSP method, but we would add this step later. And at last, we should do the orthogonal lines of W, which in this step we should get the result. And if this algorithm doesn't converge in this step, we would repeat the iteration and go to the step 4. This is original signals and the frequency spectrum of this. And this is a mixed signal using the random matrix. And this is the recovered signals. Let's do the evaluation here. We can see that in time domain it's pretty similar here, and the peaks here is almost the same. But the frequency spectrum is a little different. It's not perfect. We would improve it later. This is signal 2. And the signal 3.
We'll probably play the. We'll play the output and inputs later after this, and we have some future improvements. And since we are mix the signals linearly, so in real world, there will always be a linear mixed signal. So we probably will. If we have time, we will try to figure it out. And if you can, after this, you can hear the output. There is still noise in the output, so we may add not denoising after the separation to the outputs. And there is not a complete evaluation criteria for this, so we're planning to use correlation in this class to try to see how similar are the outputs and the original signal are. And I think that's it. As a conclusion, for this project, it's based on the similarity between IC algorithm and sampling of blind signal separation. As you can see from the pictures, the original signal are much similar as the output signals. So it pretty much achieves our goal. And I think that's pretty much it. And we will play our demo. So there's no.
I don't know if the sound. I don't know what happens with sound on this thing.
Yeah,
Very good.
there's no. Please turn on.
I don't think we can get to the sound. I don't think there's any.
Doesn't work. Probably.
Just like,
Is there a?
Maybe just play it on your laptop.
try-
Yeah,
try-
but try this one.
Yeah, this is a signal one, and this is this is what we have recovered from the mixed signal, and
And there's still noise in it.
Yeah, it's this thing. Doesn't help, right?
Last couple of days, what a custom. This is a
signal two, and
Hey, it's time for some intervention here. Step out of your car.
This is
Don't
output.
look back.
yeah, this is a signal three.
Hey, it's time for some intervention here. Step out of your car. Don't look back.
Yeah, we forgot to play the mixed signal, so maybe.
Yeah, that's.
Sorry.
All right.
Okay.
That's it. That's
All right, great, thank you. Again, we should keep moving along, so I think, right.
Sorry.
So, okay, that's lots of it, right? Yeah. So guys, I'm Nathan. Today I'm going to talk about spectral domain space microscopy. So what is it? It is a signal processing technique for optical coherence tomography. OCT is a medical imaging system using the interference of light to form images. And we want to use OCT because it performs real-time measurements, and it has a high sensitivity. And spectral domain phase microscopy is important because it's able to obtain nanoscale motion non-invasively. So if you have something if you have like a body or something, you can measure the vibration within that body without opening the actual sample. So there's a little interference graph for OCT. If you have the time, you can look at it. So what is the purpose? I work in a lab that deals with hearing. And cochlea, which is in the inner ear, is usually transduces mechanical vibration, which is sound, into electrical signal to the brain. And it is still a black box because it's kind of like a living battery. So when you have it,
when you have sound going in, it has the bending of the hair cell, which induces ions influx and then turns into voltage as a signal to the brain. But then once you open it up or you poke through the cochlea to see what's going on in there, usually it's passive, meaning it's kind of dead. You can't see the signals anymore. So with this technique, we hope to measure the vibration inside to understand what's going on in the cochlea. So I prepared this experiment by having a signal generator, speaker, the OCT system, and a membrane that is 1,000 micron with a little polymer on top. The function generator was generating at frequency from 2 kilohertz to 28 kilohertz, which is around the human hearing range. And okay, so I'm going to spare you guys the boring equations and just show you guys a flow chart. I guess you can't really see the words on here. I'll just read it out. So we have the data from the OCT that is in spectral domain, which is in wavelength. So we resample it so that it becomes in terms of wave number. Then we take the Fourier transform of that to obtain the complex A-scan. And the A-scan, I'll show you guys a picture later, it kind of looks like a peak. So it shows you where the vibration is at. So we choose the peak of that, the peak of the pixel, which is the pixel of interest, and we take a Fourier transform in terms of time, and then we can obtain the magnitude of vibration. Okay. So this is what an A-scan looks like. You can see that it peaked over here, and I obtained 3 sets of data with just the pixel at the top, and the second set is 5 to the left and 5 to the right to do a weighted average to taking account of all the pixels. Another one was 7, I think. Wait, no, 3 and 5. 3 on the left, 3 on the right, 5 on the left, 5 on the right. And then we can look at the vibration data at 12 kilohertz when I set the function generator at 12 kilohertz. We see, the most important stuff to see over here is that there is a peak over here at 12 kilohertz. And that is very important, so we can see that it's working. And then the magnitude over here is supposedly the vibration magnitude of the, make it move. Vibration magnitude of the membrane. And we can see the difference in the weighted average when we just have 1 pixel. It's slower. The DC. Oh my goodness. It's driving me crazy. Yeah, don't do that. Okay. The DC value is lower, and when you have the weighted average, it takes account of all the data, which makes it higher.
When you touch the screen, it's like clicking on the...
Oh, okay, cool. So we can see that the measured vibration, so I compared the data with interferometer at the medical campus too, and using this technique and obtain data. So I compared the 2 data, and it's kind of close. It's a magnitude away. We had to take account that it's in different environments. There might be vibration in the surroundings, and we're using different sound system. And when we have this is, I think it's 2 kilohertz. Yep, 2 kilohertz. You can see the peak here at 2 kilohertz, and weighted average is good. It's rectify certain vibrations, and it's still pretty close in terms of the data. But here, something interesting happens at 7 kilohertz. We can see non-periodic, non-sinusoidal signal here. You can see that with 1 pixel, it's kind of weird, kind of rectified, and even with weighted average, kind of fixed, but you still see a little artifact over here. So I'm still trying to figure that out, trying to see what's going on. And future work, yeah, what we're doing with durable, so that's it.
Great, thank you. Any questions for Nathan? So the successive, you're looking at a particular pixel on this
Or
scan.
it's like 1 pixel or 3 or
Okay.
5.
But and the pixels there represent a point in space, something in the
The depth, the,
okay,
yeah.
But somehow they're being successively measured. So there's those offsets that we see in 7 kilohertz data.
Hmm.
That's probably something that happened to the actual sensor between those samples or something. Or,
I'm not sure. I need to do more testing, but we just, yeah.
I mean, the point is there's a lot of processing that goes on before you can get to that point. So
Hmm. Yeah.
you're hostage to that. All right, great.
Yeah,
Thank you.
thanks.
Okay, so now it's Linda Yang and Yitan Li. Wait, hang on. Yeah. Okay. You probably won. Okay.
Hi everyone, our project is audio reconstruction from neural measurements, and my name is Yitong Li, and this is my partner Minda Yang. Okay. Our project is audio reconstruction. Is we reconstruct what we hear from the neural signals in our brain. So first of all, let's take a quick look of what the brain looks like. And brain is divided into regions in which particular functions are localized. As for us, we just focus on this part. This part is responsible for ordinary measurements, maybe. So next step is to know what's the stimulus and neural responses. When we hear something, our brain may respond to it in neural signals. For a population of n neurons, we represent response of neuron as n, neuron n and t as r of t and n. And it's also the same for the stimulus. It's worth to say that we use spectrogram to represent the stimulus. It's i's of t and f. F is for frequency and t is for time. So this is the stimulus. We hear it, and the stimulus passes through our ear to our brain, and our brain has a signal to represent it. And our aim is to use the signals in the brain to reconstruct what we hear. So here is an example. Here is the time domain frequency, the time domain signals, and we use the spectrogram to represent it. And there are a lot of neurons have response to it. So there's a question why we need to use spectrogram to represent a signal. Because a specific neuron only responds to a certain frequency range. So we need to use the spectrogram. Next step is to find the stimulus. The next step is to find the relationship between the stimulus and the neural responses. There are actually two equal equations. SF equals the convolution of G and R and times this one, because the stimulus and the neural responses, there is a nonlinear relationship between them. But the nonlinear part is trivial compared with the linear one, so we just focus on the linear one. R is the responses in our neural, and G is the function. We need to get G from our training set of data. This one is the same. It's just the inverse of the first one, so we just focus on the first one. The stimulus equals G convolves R. Here is just a different expression. We remove the nonlinear part and just leave the linear part. There's also an optimization problem because we simplify our model to a linear model. So we need to minimize the mean squared error between our estimation and the real signal. And there's a summation of t. We just need to minimize this one and get our g, and then we can reconstruct our stimulus. Here is an example of the reconstruction. Yeah, this is the solution. Or G can be get from CRR inverse and CRSF. We may skip this part and go to the result.
Thank you. Do we have speaker? Put audio play. Great. We have seen that using a simple idea as correlation, we can come up with the model for reconstruction. Next, we have seen some results. The first is the word. This is the original sound that the patient heard.
Nurse.
Yeah. And this is what we reconstructed from the neural response. I can hear that it's not very clear, but this is alignment between the signal and the neural response. You can see that when the sound is played, there is some high energy in the neural response. And next one is a phrase. This is
All right.
This is original sound, and next is reconstruction. So next one is a sentence.
Is there a pharmacy near here?
So you can see that at the end, the last word here is not reconstructed because maybe the patient is not paying attention to it. So last one is that in the previous ones, we used the data from 4 patients that they heard the same sound, and we reconstruct from the about 400 electrodes. And next, we try to reconstruct from only one brain, so only 100 electrodes. So this is the results.
Nurse.
So you see, if you use less information, then the reconstruction will be worse. So the results, average correlation between the sound and the reconstruction is 0.458, and in general, the words are better reconstructed. So thank you. Questions?
So, did you collect the data, or is it some data set?
The data is provided by University of California, San Francisco.
Okay, and this is non-invasive. It's like
Yes, it's in a surgery that the doctor will open your head and put some electrodes in the surface of the brain. That's why the data is kind of in the low quality.
yeah.
So
it's
yes, that's right. Very good point.
And so, so you said that words are reconstructed better. Is it maybe that the shorter you look, the better accuracy you get?
Yes, because in a sentence there will be correlation between the words. So this is nonlinear, and our model doesn't have that feature. So as you can see in this example,
Is there a pharmacy near here?
sorry, the last word is missing because maybe for the brain it can predict it, but our model can't do that.
Oh, really? Wow. That's great. So that's why, with a longer phrase, the brain is like, okay.
Yes, just like you listen to sentence, maybe
Yeah.
before it finishes, you can predict what it means. Yes, something like that. Very good.
All right, great. Thank you.
Thank you all.
Can I get a none? Yeah, great. Problem is when you touch the screen,
Okay.
it starts.
Oh,
When you touch that thing. Okay.
Okay.
Oh yeah.
Okay.
Okay, let me try this thing. Yeah,
Thank you.
put them- So this is the last that we got. So
Hi, my name is ELEN, and the topic of my presentation is the studies of features of music instrument waveform. In my studies, I took the four type of music instruments: violin, piano, flute, and trumpet, and I take the measurement of their waveform, and I think, as we know, for this four type of musical instrument, it is pretty easy for us to tell the difference by just listening to them. And when I plotted the waveform frequency response, it is also easy to find out the difference. But the one big challenge here is to figure out what features we can use to quantify the similarity and the difference between the different type of musical instrument. And here are some features that are proposed here. It might be useful, might be not useful, just interested. And here are the samples. These are actually the sound sample got from the website. So for each type of music instruments, there are 6 or 7 different type of notes here. When I started the project, the first thing I'm thinking is there any similarities between different musical instruments at the same note? And the answer is yes. The first thing I found out is if we take a look at those waveform frequency for the 4 type of music instruments, the first spike here, the wavelength for the first spike is always the same, and that is actually corresponding to those numbers here on the website. And the second thing I found out is at the same notes, the number of the major spikes here are the same. And the frequency for these spikes is also very close to each other. And the spacing between these frequencies is also pretty same. It's about 200 hertz. And after that, I'm trying to compare with those waveforms of these 4 type of instruments. And this is, I did a combination, combine all the notes together for every instrument. So this will be the violin from notes C4 to G6. So by looking at these waveforms, I say the piano has a much different waveform from the other 3 because this baseline here, or we can call it a noise, it's decreasing, while the others looks similar, horizontal. And the flute is also special. I found at the higher frequency, the spikes decreasing very fast, much faster than others. And at higher than 40,000 hertz, there's almost no big spikes here. For the violin and the trumpets, they are very similar. But one difference is if we look at the spikes here, the spikes of the violin looks more coarse than the trumpet. And then I came up with several features to try to describe these differences with a certain number or quantitatively. And this one, the spikes, is the most important, most critical features I found, because other features are also closely related to this feature. So first, I give the definition, some definition on the spike here. What I'm interested is those major spikes here, but if I zoom it in, there's a lot of small spikes, mini spikes here. To be a spike, first it must meet the requirement that is a local maximum. That means it's higher than the left one point and the right point. And also, I don't want to take into account of the many specs here. So at a certain range, I only counted the one with the highest maximum. And the third requirement is the magnitude has to meet some requirement. This is mainly to eliminate those spikes at a very low level here. Because if I just meet the first and second requirement, those noise may also be taken into account. That is not what I want. And at a frequency higher than 4,000 hertz, those spikes have a pretty lower amplitude. And I also want to take this into account. That's why I have the different requirement of amplitude for the higher frequency range. This is another feature called cutoff frequency I defined. This means the frequency when the spike magnitude goes below the 0 dB. And by comparing these 4 different instruments, I found the fruit has a cutoff before the 4,000 hertz, and the other 3 doesn't have. This one looks quite tricky because this looks like it has lower than 4,000 is too. But if we take a look back of the piano signal, the actual magnitude of the spikes is actually big. It is lower than 0 because its baseline is decreasing. So I won't take the piano into account for the cutoff at 4,000 frequency. So this actually tells a major difference from the fruit and the other 3 components, other 3 instruments. Is that the fruit has a much higher decaying rate and the cutoff frequency at 4,000 K or 4,000 hertz. And the next is called the full width half maximum of the. Basically, it means the width of the spike. So I measure the width of the frequency when the amplitude going to 50%, and compare with those 4 instruments, and I found the violin has actually the result is pretty interesting. I found the violin has the highest width, and the piano has the very smallest width. And the trumpet and the flute has a very similar width. Even when I combine the different notes together, they are still pretty similar. So I suspect, perhaps because those both trumpet and flute are both played because of the airflow mechanism. So that's why the width is very similar. And combining with the finding I introduced before, for the different instruments, the wavelength location of the peaks is very similar. So if there are 2 instruments have the same frequency location but a different width, then we can very easily develop a certain filter, like a band pass filter or some filter to filter out one type of signal and keep the other one, like violin and piano. And next is the fluctuations. Here I'm counting the small spikes in near the main spike region, which is plus minus 40 hertz. And this I didn't find any clear change by comparing the different notes. I didn't find anything special. The only thing special is the trumpet has more spikes compared with the other, but it may not be always the case. And the last feature is the dB line crossing distribution. This one is merely to show how the baseline varies. So I developed a certain group of dB lines ranging from -60 to 20 dB with 2 dB as an increment, and counting the crossings of the waveform to this magnitude, and I plotted the distribution, the number of crossings as a function of the dB length. And quite expectedly, the piano has much more scattered distribution compared with the other three. And I think this is probably because if we take a look previously, compare these four different plots, the piano has more sparse, the baseline distribution have a bigger range compared with the other three. And also, the piano looks more sparse, and the waveform is not very concentrated. So the actual number of piano crossings looks smaller than the other three. And here is some future possible studies I think we can do in the future. One is the decaying rate of the spike. The spike of the shape of the spikes looks very different as well. Like the traffic has a certain shape for every spike here, but the other instrument doesn't have. And this is the one I mentioned earlier. We can use the the width feature and the position of frequency feature to differentiate the violin and the piano signal, because they have very big difference in the width, but the same location. And also another thing maybe we can do in the future is to remove the noise and only keep the specs and see what it sounds like. And I think these features may also be helpful for analyzing other type of instrument, even when they have a very similar sound output. And trying to use those features to separate those mixed signal. That's it.
Great, thank you. Very nice. All right. Yeah, we should move along. That's great.
Okay.
And
I don't need a mic. How you doing, everybody? My name is Preston Conley. This is Maya Rudolph. That our project is about walking pace extraction coming from video. So here's a overview of what we're going to be talking about. We talk about our goals, which is to find out the average speed from trying to find the average walking speed from a person walking in a video. We're going to talk about like our approach, which is an approach is like the assumption we are assuming that every person has the same stride length while walking, and also we be talking about what process we do to figure out the stride frequency which is k-means clustering. We'll go over our results and also our future work. So that just as I said previously, our goal is to abstract somebody walking, because here we have a 3-second video where while a person is walking, that we are trying to since it's a periodic motion, so we're trying to exploit that property, and so from that periodic motion we can find the stride frequency, and then multiply that by the stride length, we are able to come up with the average speed. And then we found the average stride frequency by k-means clustering.
Yeah, and so how we did that, I will tell you a little bit about it. So would you mind to flip the pages for me?
Sure.
It's what with
Yeah.
the arrow button.
Yeah.
Yeah, and so we can go to the next page. Right? So each frame in the video, we wanted to exploit that there's somewhat a periodicity, that at some point the legs are closed, then the legs are open, and so forth. And we wanted to see, like, cluster the frames by similarity. And we did that with k-means clustering. So each frame, it can also be seen as a 160 times 120 times 3-dimensional vector. 3 is because there are 3 channels for the 3 colors, red, blue, and yellow. And so, red, green, and blue, sorry. And then, so in this high-dimensional vector space, we just took Euclidean distance, which is like the most straightforward thing to do k-means clustering with. And so with k-means clustering, you specify how many clusters you want. And using cross-validation, we discovered that 3 worked really well. So we fed those 113 frames into k-means clustering. And here plotted, you see the cluster centers, which are the average of the frames that were assigned to the clusters. And this was really exciting because we actually had this legs open, legs closed frames as we expected. And here you can see a plot of, on the x-axis you have the frames, and this is the distance of the frames to the specific clusters. And so when we saw these plots, we got really excited because you actually exactly see this periodicity. Like here, it's really close. The clusters are really close to legs open, and they're really close to this other position. And so now all that's left to do is to exactly extract this periodicity of the strides. And so there's one other very straightforward plot, which is the cluster assignment. So each frame got assigned to a cluster, and so we see here is cluster 2, then it's cluster, I don't really see it right now, but it's like alternating between cluster 1 and cluster 3. And so really what we want is like, how long does it usually take when it was in a cluster to come back to this cluster? And this would give us the length of one stride. And so we did this with a very straightforward counting, and then we extracted, like we also used some threshold to throw out stuff that was a little bit away from the average, and then we averaged again to get exactly this periodicity, and then it came out to be maybe, yeah, exactly 13 frames for a step, and then we could calculate, okay, there are 8.4 total strides with this method, and that exactly coincides with counting. Like we just counted how many strides are there actually, and so this method worked. And with the assumption that the stride lengths are constant, we actually got the velocity. And yeah, next slide. And so we also tried this with other videos. So we used very straightforward feature where each pixel gets assigned to the pixel, but if the person would actually be moving in the video, this wouldn't work anymore. So we would have to use other features. So this could be done using optical flow or a different distance metric. And we could also try to restrict the clustering to body position to avoid like noise in the background. Maybe there's a car in the beginning and then there's no car in the beginning, and then this is what discriminates the clusters. And also, instead of k-means clustering, we could use hidden Markov models, which additionally to putting similar stuff together also take into account what the frame before and after a frame, which cluster they were assigned to, which also makes sense because it's like it's a temporal dependency there. And yeah, we're really excited to explore this further, and thank you for listening.
Any questions for Preston and Leah? So you had that nice result where the 3 clusters fit very well. What would it look like if you used different numbers of clusters?
So with bigger numbers, we also got that there were always those 2 clusters which had this really nice periodicity,
Yeah.
but some like noise clusters. But unfortunately, it didn't work with 2, and my guess is that the person is just not centered at the same place in the beginning and the end. And that's why they were, so if you remember the plot there, it was like cluster 2, and only in the middle it was nicely alternating. So that's why we decided to use 3. Yeah.
And so the critical thing about that one video is that the camera pans with the figures, and how many videos can you find like that, where the person stays fixed?
Yeah, so for example, in like sports races, when they're filming sports races, then it's also following the guy. But then, for example, if you would actually track the body using optical flow, seeing how much each pixel is moving, then you can associate
so you can actually center it afterwards. You sort of use optic flow to say what stuff is happening, and then I mean, in theory.
You don't really need to center it anymore, but then you can see how much the pixel changed and just define a distance metric on that for your clustering.
Great. All right. Thanks a lot.
Thank you.
Okay, so now we're doing great, and now we have our last talk, which is Alan, Tanya, and Sophie. Great.
All right, so we're gonna be talking about the classification of lung sounds, and let's start off with something we're all really familiar with, which is the physical exam. Most often, when you go to see a doctor, they listen to your lungs. You take a few breaths, and they're listening for these specific sounds, which 2 of the main ones are called wheezes and crackles. This is really commonly used. It's never a final diagnostic tool, but for example, when you first come in for a physical exam, it's used, and then it's also used to monitor the progress of disease. The problem is, although it's used all the time in the hospital, it's not really objective, and there's no real way yet that's used to quantify this. Also, even after many years of training, doctors hear different things, or they're not sure what they hear, and what ends up happening is they prescribe either antibiotics preventively, or they send people for x-rays when they shouldn't be doing this. There's a huge high positive rate. So what they're really listening for is the frequencies of specific sounds and their durations. Some sounds are longer and some musical. Some sounds come in really short high-frequency bursts. So just to give you a sense of what it is, here's a typical wheeze. Let's see how many figures. All right, let me play one more time, sorry. And then it's just really common in patients with asthma, for example. And then this is a crackle. And you can hear the crackle. Here it's pretty easy to distinguish those 2. But then in something like this, right, it's much more difficult. And so the problem is, most often you get something that's in between, or there's a lot of noise. Maybe the stethoscope is running against the patient's skin. There's ambient noise. It's a loud hospital. A ton of problems come up, and the doctors don't really know what to do, so to be safe, they send for an x-ray. So we wanted it to sort of, this is how we broke up what we were trying to do here. We took online sound banks acquired with electronic digital stethoscopes, and then looked at these signals for things that we know are crackles and wheezes. We analyzed them in the time and frequency domains, split them into two groups, one to train and one to test, and then decided on features which are going to be characteristic for each of the sounds. Based on those features, we built a classifier, and then saw how they performed on our training and testing data. But basically, right after the first iteration, we realized that there's some of the microphones have sort of a thresholding that cuts off the sound at certain points, and there's sometimes long times between breaths. So we split up the sounds of multiple breaths into single breaths, and increased the power of our analysis because we have more data points, samples to deal with, and also took away some errors. So basically, here's this is the time domain visualization. Here's a wheeze, and this is a crackle. You can't really tell the difference, but as soon as you take it to the spectrogram, you see that there's this really wide frequency band that's characteristic of crackles. And there are these short bursts, while wheezes stay below 2,000 hertz. And so we tried to sort of look at which features we're going to emphasize here to extract and eventually classify. The problem is when you get a spectrogram, it doesn't look that pretty. It starts off like this. So we applied this mask that is basically a filter that changes at every time step, meaning that it would take away all the frequencies of intensity below negative 25 decibels. Through trial and error, we sort of figured out that this would be the ideal way to take out the part of the signal that we really want to see.
So based on those spectrograms, we decided to look into frequency and the distribution of energy across the frequency spectrum as a potential feature to extract and discriminate between the two classes. So we looked at the energy distribution across frequency, and these are averages across time and across samples. And so here on the left, you have the distribution for crackles, and this is a distribution for wheezes. And as you can see, there are peaks in the high frequency spectrum for crackles that you don't see in wheezes. And what we wanted to do was to analyze how the energy is distributed across the spectrum, and so we took a ratio between the energies in the high frequency band and divided by the energy in the low frequency band. So through a lot of experimentation, we determined the optimal frequency band for the high frequencies would be from about 1500 to 5500 to capture all of that energy there. And then the low frequency spectrum was much narrower, from 400 to 500, and this was supported by literature which showed that wheezes had pretty characteristic frequencies around 400 hertz. And so we characterized it and quantified how well that worked by looking at plots of these ratios between these two classes. And we can see there's some separation. We see that the crackles have higher ratios as we would expect, but it's still not perfect. And we looked at a ROC curve to kind of see how this discriminator, how well this discriminator actually discriminates between the two classes. And the area under this curve actually represents sort of the accuracy. And it's a metric that we used throughout the entire development process. And here we saw that it was a 78% accuracy rate. And so while that's not great, we thought that maybe adding more features would actually increase the power of this discriminator. And so the second feature that we looked at was the temporal variance in the frequency bands. Since, as you heard in the sound samples, the crackles are much more burst-like in frequency, whereas the wheezes are much more continuous sounds. We looked at the variance in energy across the frequency spectrum. And we saw, again, a peak here in the high energy part of the spectrum, in the high frequency part of the spectrum. And so we decided to use the variance in that same band that we described before, from 1500 to 5500, as our second feature. And so here's another plot comparing the two variances across the two classes. And here's our ROC curve, and our accuracy here was about 63%. So while that's not as high as for our first feature, we thought the combination of the two would increase our classifier's power.
So we also looked at a third classifier that we could use. LPC, or linear predictive coding, basically predicts future values from a linear function of past samples. And you can subtract that predictive signal from the actual signal to get an error differential that you can use to classify. We thought that because crackles had high frequency spikes, that there would be a larger error with crackles that we could use to differentiate between the two. However, as you can see from the mean squared error plot, there's a lot of crossing over between crackles and wheezes, and the ROC curve gave an A value of about 62%. While this is the same as the variance A value, the time required to do this program was like 24 times larger than the other two combined, and so really increased our time without increasing the power of our differential, so we decided not to use it in the resulting analysis. And so we took our two features that we decided to use and put them on two different axes to get a two-dimensional space. We used training data, 25 samples from each class, to create a linear classifier. You can see that with the green line right there. And blue and red represents the training for the wheezes and crackles, we then used testing data from both classes to check how well our linear classifier worked. For the crackles, we got a 100% success rate. For wheezes, we were about 78%, which ended up giving us an 80.5% success rate as a whole. Though these results are pretty nice, there's some limitations that we really need to consider when looking at our implementation process. For one, as we talked about in the beginning, a lot of samples are a mixture of crackle and wheezes, and so our binary way of classifying might not necessarily work for those kind of samples. We also didn't have control over our method of acquisition because we took samples from the internet, and so different acquisition methods might have changed the way that our analysis worked. In addition, we had a limited data set. We only had 5 crackles that we tested on, and so if we had had a greater number of crackles, it might have changed our results or enhanced it in other ways. So for future directions, it would be interesting to look at other types of pathological long sounds. We chose crackle and wheezes because those are the two most common that appear in physical exams, but there are also things called striders and squawks and other ones that we could try and classify using a similar method. We also want to try and look at wavelet, or we might want to look at wavelet analysis for better temporal feature extraction. In addition, there's artificial neural network classification, so there's a couple different classification schemes that we could look at if we had more time. In conclusion, as we can see from the frequency analysis and the temporal analysis, there are differences between the two major pathological long sounds, crackles and wheezes, and it's just a matter of enhancing those features and choosing the right features to assess those. We found that the spectrogram was absolutely essential for analysis because it really showed the differences between the two. When we looked at just a frequency plot, you couldn't really see differences between crackles and wheezes. And so basically the discrimination hinges on the strategic feature extraction. And so picking the right features, making those, or weighing them correctly, all of that. We also found that the entire process was completed in a clinically useful time. So per breath, it was about 14 milliseconds to get a result, which for a minute of acquisition time is about 1 second for the program. So it could be useful for physicians.
So,
Thank you.
all right, excellent. I think we're going to have to wrap it up there because we ran over a bit. But thank you for everyone. Those are really nice presentations, really interesting. I hope you all enjoyed that. So that's a really nice way to wrap up the class. We'll be meeting here on Wednesday. If you want to show up, I'll just be doing a review, going through some review slides of the second half of the semester and answering any questions you may have when preparing for the final exam. Final exam is here on Monday at 9 o'clock, so please be here. And then the nominal project deadline report is due also on Monday by the end of the day. Preston.
Is it open notes?
Yes, the exam is the same as the midterm, so it's open book, open notes, but only calculator, no laptop. All right. Okay. See you again.